In this video, we discuss about the topic enzyme that's come under microbial physiology metabolism, that is B.V.MLT 303. Okay. So first of all, what is enzyme? Enzyme is nothing but it is only a protein. All enzyme are protein. Remember this. This is a common statement. All enzyme are protein except remember this, except ribozyme. Okay. All enzyme are protein except ribozyme. Enzyme help act as a vector. Enzyme help in increase the rate of reaction. What? They increase the rate of reaction. They decrease the activation energy. What? They decrease the activation energy. If some reaction takes place, suppose A react with B, right? And they form AB. During this process, suppose without enzyme, this process takes place in 20 minutes. When A plus B, A react with B in presence of some enzyme, suppose this is enzyme, and form a AB product, when enzyme is present, they complete this reaction in 2 minutes. What? They complete the reaction in 2 minutes. So what happened? Rate of reaction will be increased here. Without enzyme, this process will take place 20 minutes. With the help of enzyme, this process will take place in 2 minutes. Suppose we want to go from Delhi to Agra. Right? From Delhi to Agra. Without any vehicle, when we go, that time takes place, how much time takes place? When we go by walking, 2 days. 2 days it takes place, right? And when we takes a vehicle, like a car, motorcycle, anything, not a flight, car, then what happened? We go in 4 hours, 5 hours, like that. So what happened? When we take something, then what happened? The duration of the time will be decreased. We go earlier, right? Same thing happened with enzyme. They increase the rate of reaction, they decrease the time, or they decrease the activation energy. Okay, clear this? So, what is enzyme? Enzyme are a protein, already I told you. Except, enzyme are protein, except, remember this, except ribozyme. All enzyme are protein except ribozyme that increase the rate of reaction by lowering the energy of activation. By lowering the activation energy. They catalyze nearly all the chemical reaction taking place in the cell of the body. They not altered or consumed during the reaction. Remember one thing. Enzyme not enter in the reaction. Suppose when A react with B with the help of enzyme, they not alter this reaction. They only increase the rate of reaction, but they not go itself inside the reaction to combine with AB or form another product. That will not take place. And we again, after the completion of this reaction, this enzyme will be reusable. What? This enzyme will be reusable. We use this enzyme. Understand? This enzyme will be reusable. So what happen in enzyme? All enzyme are protein. Remember this, all enzyme are protein except ribozyme. They catalyze all the chemical reaction takes place in the body. They not altered or consumed during the reaction, and they are reusable. We can use again after the reaction process. Enzymes share property with chemical catalysts. Enzymes are neither consumed nor produced during the course of reaction. What? Enzymes are neither consumed and nor produced. They remain as same. How we insert the enzyme during the reaction, how we put the enzyme during the reaction, they remain the same after the reaction. They not change their property after the reaction. Okay? Enzymes do not cause reaction to take place, but they greatly enhance the rate of reaction. What? They greatly enhance the rate of reaction that will proceed much lower in their absence. They alter the rate but not the equilibrium constant of the reaction that they catalyze. Remember this. The enzyme enhance the rate of reaction but not alter the rate of equilibrium. Equilibrium. They not alter the rate of equilibrium. Clear? What is the difference between the enzyme and chemical catalyst? Enzyme are protein. Enzymes are highly specific and produce only the expected product from the given reactant or substrate. There are no side reaction with the enzyme. Right? Enzyme may show a high specificity towards one substrate or inhibit a broad specificity using more than one substrate. Suppose this is an enzyme and this is an active site. Already in another slide, what is active site, what is the substrate site, we already discussed in later, right? This is an active site, right, where substrate come and bind. So one enzyme, remember one enzyme is specific for one substrate. One enzyme is specific for one substrate. Enzymes are highly specific and produce only the expected product from the given reactant or substrate. Enzyme may show high specificity toward one substrate or exhibit a broad specificity using more than one substrate. Enzyme usually function within a moderate pH and temperature range. All enzyme will maximum enzyme will work in moderate pH and moderate temperature. Right? Means the pH will be 7.4 or temperature will be 37 degrees Celsius. All enzyme will be work at this pH and temperature. When the free energy of the reactant is greater than that of the product, the free energy changes for the reaction is negative. So the reaction proceeds spontaneously. If delta G that is free energy is negative, means reaction with spontaneous. If delta G will be positive, means reaction is non-spontaneous. Right? When the free energy of the product is greater than that of a reactant, the free energy changes of the reaction is positive. So the reaction does not proceed spontaneously. They required some extra energy. Right? When the free energy of reactant is more as compared to product, then this process is known as spontaneous process. When free energy of the reactant when free energy of the reactant will be less than product, when the free energy product is greater than that of the reactant, free energy of the product is greater than that of reactant. The free energy change of the reaction is positive. Delta G will be positive, and this is a non-spontaneous, right? And this is spontaneous because delta G will be negative here. So remember this. What happened? This free energy will be depend on the reactant and free energy of the reactant and the product. Okay? Clear? Enzyme lower a reaction activation energy. What happened here? Enzyme lower the, what happened? Enzyme lower the activation. How they lower the activation energy? All chemical reaction require some amount of energy to get them start, right? All chemical reaction require some amount of energy to get them start, or it is the first push to start reaction. This energy is called activation energy. What called? This energy is called as a activation energy. See, this is a ball, right? For this ball, when this ball go inside, for this ball transfer one side to another, they require some energy. We push this ball, right? When pushed, we push this ball go in another side, right? So when pushing the ball, some energy will be consumed. That energy is known as activation energy. What? That energy is known as activation energy. This is lactose. This lactose break into a glucose and galactose without enzyme, right? So in this process, high amount of activation energy will be required. And when with the presence of enzyme, when lactase is present, this enzyme will break into glucose and galactose easily. Less activation will be required. So in this process, when we push this ball to there, and when we down the slope, what happened? Less energy will be required, right? Less energy will be required, and that is the activation energy. So less activation energy will be required here. This is the activation energy without enzyme. No net energy released from splitting of the lactose. And here what? This is activation energy with enzyme, net energy released. So what happened? Activation energy will be low when enzyme will be used. Clear? When we use the enzyme, the activation energy will be low. This is the diagram of the how the reactant and product will be react, and how the activation energy will present in presence of an enzyme and in absence of enzyme. This is a reactant. When reactant will convert it into a product without an enzyme, course of reaction without an enzyme, the activation energy will be more. And when this is a course of reaction, red color, in the presence of enzyme, when the reactant converted into a product, course of reaction with enzyme, right? This is a activation energy without enzyme. They require more or they require less. Delta G is unaffected by enzyme. There is no change in delta energy, because delta energy is calculated by the product minus reactant. Delta G of product minus delta G of reactant. So there is no change in delta G. There is only change in the activation energy. The energy will be low. What are the components of the enzyme? The main component of the enzyme is apoenzyme and coenzyme, right? This is the apoenzyme. What? This is a apoenzyme. When apoenzyme combine with coenzyme, apoenzyme when combined with coenzyme, that called as holoenzyme. Remember this. When apoenzyme combined with coenzyme, they called as holoenzyme, and this holoenzyme combined with substrate, right? What is coenzyme? Coenzyme is a factor. What? Factor, cofactor. Cofactor like they contain metal, vitamin, mineral, which increase the rate of reaction. The substance will increase the rate of the reaction. And what is the apoenzyme? Apoenzyme is a protein molecule. What? Protein molecule. Coenzyme is a cofactor like metal, vitamin, which enhance the rate of reaction, and apoenzyme is a protein. Okay. And when combined with this, both apoenzyme and coenzyme, when protein will be combined with the cofactor, that call as a holoenzyme. And that holoenzyme bind with substrate, right? Now, what is a cofactor? What is a cofactor? Cofactor is an essential ion and coenzyme. What? The essential ion and coenzyme. Essential ion are two types: activator ion and metal ion or metalloenzyme. Activator ion, they bound loosely, only they activate the process of the enzyme. And coenzyme, a co-substrate or prosthetic group. In co-substrate, they bound loosely bounded, and prosthetic group bound tightly bound. Okay. Remember this. Cofactor are of two types: essential ion and coenzyme. Coenzyme are of two types, that is coenzyme contain co-substrate and prosthetic group. Prosthetic group bind tightly. This is a enzyme cofactor. What? This is a enzyme cofactor. Activator, prosthetic group, coenzyme. Prosthetic group bind tightly. This is a non-protein part. Already remember that coenzyme cofactor is a non-protein part. Cofactor itself is a non-protein part. Protein part is a apoenzyme. So, what is prosthetic group? Prosthetic group is a non-protein part of the molecule such as heme group, porphyrin or catalase, right? And coenzyme are a non-protein part usually derived from vitamins, which consist of enzyme in their function. They are physically separate from enzyme, right? This is the enzyme and this is the enzyme activator, that is a cofactor. When they combine with this, they called as a holoenzyme, and this holoenzyme bind with a substrate and increase the rate of reaction. Activators such as chlorine ion in saliva activate salivary enzyme, and calcium ion activate thrombokinase in blood clotting. Okay, remember this. What is a cofactor? Cofactor is not a protein part, it is a non-protein part. This is a structure of enzyme. This is a whole enzyme. Here there is a site where substrate come and bind. This site is known as active site, and here prosthetic group will be present. Prosthetic group will be attached there, which increase the rate of the reaction, FAD, right? This is the enzyme, this is active site, this is a coenzyme will be present, will increase the rate of reaction. When reaction will, here it is attached and here it is detached. Okay. So, coenzyme and prosthetic group is a non-protein part. This green color is only a protein part, green color, only green color. Green color is a protein part and this yellow color is non-protein part. This green color is protein part and this red color is non-protein part. Okay. When these both are combined, they form a holoenzyme, and this holoenzyme combined with the substrate increase the rate of reaction. Clear? Now what is active site? Active site of area in the enzyme where substrate come and bind. Active area on the enzyme where the substrate or substrate attaches to is called the active site. Okay. This is a active site is a binding site. They hold the substrate in place. And this is a catalytic site. What? Catalytic, where reaction occur. This is the active site, this is a catalytic site where reaction will be occur. And this is a substrate. This substrate go and bind with this. And reaction will takes place. Okay? Clear this all? See here. Substrate will be entered in the active site of the enzyme. This is the active site. Here substrate come and bind. Enzyme substrate complex will be formed here. Enzyme changes shape slightly as substrate bind. What they called enzyme when substrate will be bind to this active site. Suppose this is the substrate. This substrate come and bind with this active site. What happened? They change in conformation. What? They change in a conformation. Conformational change will be occur. Right? See here. Enzyme changes the shape slightly as substrate bind. Shape will be changed. This is enzyme product complex. When conformation will be changed, this substrate will converted into a product. Substrate will converted into a product. Now what happens when product will be formed? This product will be detached from the enzyme. This product will be detached from the enzyme. Okay? Clear? When the substrate will bind with the enzyme, their conformational changes will be occur. Right? Conformational changes will be occur and they converted into a product. After converting to product, the product will be released and enzyme will be remain as same. The enzyme and substrate bind with each other with the help of bond. That is weak bond. What? That is a weak bond. They may be hydrogen bond, van der Waals bond. Okay? They attached with weak bond, so that they after converted into product they easily separated. This is the enzyme, this is the substrate. They combine, form an enzyme substrate complex. Conformation change will be occur. They convert it into product. The product will be released and active enzyme will be released. This is enzyme. This is a substrate inhibitor. They inhibit the enzyme, and here inactive enzyme will be formed and no product will be formed here. This is a substrate inhibitor. This is a substrate inhibitor. This is enzyme. Right? Same case here also. They inhibit the enzyme. They inhibit the enzyme. After inhibiting, conformational changes will not occur, so that enzyme will be inactive here. So, inhibitors are also present which inhibit the activity of the enzyme, so that enzyme will be not work properly. And reaction will be stopped there. What are the theory of the enzyme kinetics? First theory is lock and key model. First, what happened in lock and key? You know that what happened in lock and key model. Lock and key means Lock and key means we know that every lock will be open and close with the help of key. What? With the help of key. Suppose this is a lock. Suppose this is a lock. Right? This is a lock. Right? This lock will open with the help of key. Right? With the help of key, this lock will be open or close. So according to that lock and key model, enzyme will be work. This is a enzyme and this is the active site. This substrate will act as a key. This substrate will act as a key. When substrate will bind with this active site, the enzyme will become active and conformational change will be occur and they convert the substrate into a product. And this active site will specific for their substrate only. Not another substrate will come and bind with this active site. No. This will not happen here. So in the lock and key model of enzyme action, the active site has a rigid shape. The active site has a rigid shape. Only substrate with the matching shape can fit. The substrate is a key that fit the lock of the active site. The substrate is a key that fit the lock of the active site. This is an older model, however, and does not work for all enzymes. Actually, lock and key model is the first model for the enzyme. It is the older model. But this model is not work for all type of enzyme. But for maximum enzyme, this model is work. Lock and key model. Clear? Another model is induced model. Induced model means substrate will be induced in a active site. After that, conformational change will be takes place. Conformational change will be takes place, and substrate will be converted into a product. Right? In the induced fit model of enzyme action, the active site is flexible. What? In this model, active site is flexible. But in lock and key model, active site is a fixed type, not flexible. That is rigid type. Means only that substrate will come and bind. But in induced fit model, the active site is flexible. They are not rigid. The shape of the enzyme active site and substrate adjust to maximize the fit, which improve catalysis, There is a greater range of substrate specificity. This model is more consistent with a wider range of enzyme. Because in this model, the active site will be flexible, so that this model is accepted with the wider range of the enzyme. In this model, scientists say that enzyme will be flexible. Substrate will come and bind according to their flexibility. If substrate need more space, the active site will be increased. Right? The active site will be increased in their shape, so that substrate will bind in this active site and they convert it into a product. Okay. Next is classification of the enzyme. Enzyme are classified into 7 groups. First is oxidoreductase, second is transferase, third is hydrolase, fourth is lyase, fifth is isomerase, sixth is ligases, and seventh is translocases. Oxidoreductase means oxidation and reduction. Oxidation reduction with enzyme which help in oxidation reduction reaction. Transferase means the enzyme will help in the transfer of the one substrate to another substrate. Hydrolase is breaking of the water molecule, like that, hydrogen molecule. Liases is also help in breakdown. Isomerase conversion of one structural form to another structural form. Ligases joining. Translocases means translocate the one place to another place. Translocate the substrate from one place to another place. How the enzyme will be recognized? Because in till a date there are so many enzymes are present and number of enzymes will be discovered. So how we recognize the enzyme? Which enzyme is this and which enzyme will be that? How we recognize? The enzyme will be recognized with their EC number. That is enzyme commission number. What is EC number? EC number is the enzyme commission number. Is a numerical classification scheme of the enzyme based on chemical reaction they catalyzed. Right? This EC number will classify the enzyme according to the chemical reaction catalyzed by the enzyme. Clear? The chemical reaction catalyzed is the specific property that distinguishes one enzyme from another. EC number specify enzyme catalyzed reaction. What? They catalyze the enzyme catalyzed reaction. Specify the enzyme catalyzed reaction. EC number are assigned by the nomenclature committee of the IUBMB, that is the International Union of Biochemistry and Molecular Biology. According to the EC number, all the enzyme will be classified. Next is systematic nomenclature. Every enzyme consists of a code of the letter, same as EC number, followed by 4 numbers separated by period. The first digits define the general type of the reaction. In EC number, 4-digit number will be taken. The first digit define the general type of reaction catalyzed. Means first this specify the types of reaction catalyzed. Right? Second figure indicate the subclass. Third figure indicate the subclass. And fourth indicate the serial number of the enzyme in the subclass. What? They indicate the serial number. When they discovered. They indicate the serial number when they discover. Right? So, these are the EC number 1, 2, 3, 4, 5, 6. First is oxidoreductase, transferase, hydrolases, liases, isomerase, ligases. Oxidoreductase, suppose this is the EC number 2711. This is the EC number 2711. Each enzyme has classified number consisting of 4 digits, that 2711 means 2 is for what? How, for which reaction they are specific. Right? Suppose this is a hexokinase, means this is oxidoreductase or transfer. 2 means transferase. Then 7 means they are subclass having transferase subclass. Then 1 is, after that they are subclass, and 1 serial of their discovery. Okay. Next is isozyme. Within an organism, more than one enzyme may catalyze a given reaction. That multiple enzyme is known as a isozyme. What? Multiple enzyme are known as a isozyme. If a reaction, suppose A plus B converted into C, this reaction will catalyzed by more than one enzyme. 1, 2, 3, 4, 5. This all are enzyme and this all catalyze this reaction. Means this 1, 2, 3, 4, 5 are a isozyme. What? These are a isozyme. Isozyme differ in their catalytic property. Consequently, the various isozymes that are present in different tissue or at different developmental stage can carry out slightly different metabolic function. These are a isozyme. Okay. This is substrate concentration and reaction rate. Already you know that when enzyme will present, they increase the rate of reaction, right? They increase the rate of reaction and substrate are specific for their enzyme. Suppose this is enzyme, right? And this is a substrate when present. They increase from this, and this is a maximum where substrate will be bind. Maximum substrate will bind with the enzyme. After that, they decrease. Means here the concentration of enzyme will be low. After that, concentration of the enzyme will be low. This and the velocity of where the maximum enzyme will bind their substrate, this is known as Vm, maximum velocity. Clear? This is known as Vm. Next is regulation. Enzyme activity can be regulated. That is, enzyme can be activated or inhibited. Enzyme can be activated or inhibited so that rate of the product formation respond to the need of the cell. If you want to decrease or activate or inhibit the level of enzyme, you can add some cofactor so that they inhibit or activate. So we can regulate the enzyme. Measure of enzyme activity. Enzyme activity can be measured by two factors. First is a specific activity and second is turnover number. A specific activity is usually expressed as a micromole of substrate transformed to product per minute. Remember this. What? Per minute milligram of enzyme under optimal condition measurement. Remember this micromole, this unit micromole, and this is per minute. Remember this. Micromole per minute. This is very important as unit of the specific activity. Next is turnover number. That is kcat is the number of substrate molecule metabolized per enzyme molecule per unit time. Unit is per minute or per second. How much enzyme catalyze the substrate in 1 minute or in 1 second? That is known as turnover number. Clear? This is the graph of Vmax, Km, this all. Okay? This is the graph. Already I told you. This is where the maximum enzyme will bind their substrate. This is known as Vmax, and Km where the half of the maximum velocity. This is known as Km. Right? When we draw a graph, the reverse of Km is the minus 1 by Km. Next is in types of inhibition. First is competitive inhibition. In competitive inhibition, the inhibitor will be bind at the active site. Suppose this is the inhibitor. This inhibitor will bind at active site where substrate will bind, and this process is known as competitive inhibition. So that substrate will not bind with that active site, and the whole process will be inhibited. In competitive inhibition, the inhibitor bind to the substrate binding site as shown, so that those preventing substrate binding. Competitive inhibition causes the Km value to increase but does not affect Vmax. Right? Clear? In non-competitive inhibitor, inhibitor will bind another site of the substrate. Means in this process, inhibitor come and bind with another site of the substrate. After binding with the inhibitor, the reaction will be stopped. So this process is known as non-competitive inhibitor. So this is all about the enzyme. Okay, hope this is clear. Okay, thank you.